All right, let's see how sharp you really are. 10 questions, no hints, no second chances. I'll give you the statement. You decide, fact or fiction. Think fast, trust your instincts, and keep score because this one gets tricky. Ready? Let's test your knowledge. Question 1. Here's your first statement. Scientists have recovered flexible, vessel-like structures from inside a Tyrannosaurus rex bone. The answer is fact. In 2005, researchers announced an extraordinary discovery inside fossilized T-Rex bones. After carefully removing the hard mineral portion of the bone, they uncovered transparent, branching structures that resembled blood vessels. Some of the material was surprisingly flexible and could even return toward its original shape after being stretched. But this does not mean scientists discovered fresh, unchanged dinosaur blood vessels. The structures had survived millions of years of chemical alteration. Researchers described them cautiously as vessel-like structures containing evidence consistent with remnants of the dinosaur's original tissues. Why does this matter? Because it challenged the long-held assumption that every trace of biological material must disappear after only a few million years. Question 2. Next statement. When paleontologists say soft tissue, they mean fresh dinosaur flesh that is still soft to the touch. The answer is fiction. This sounds believable, but it is not accurate. In paleontology, soft tissue usually describes biological material that was not hard or heavily mineralized when the animal was alive. That includes skin, muscles, organs, feathers, blood vessels, and cellular structures. The fossilized material does not need to remain soft. It may survive as a thin carbon film, a mineral replica, a microscopic chemical residue, or a structure transformed and stabilized during fossilization. So when you hear that dinosaur soft tissue was discovered, do not imagine a piece of fresh meat. Imagine an ancient biological structure that has survived through an extraordinary combination of chemistry, burial conditions, and time. Question 3, let's continue. Amber is the only material capable of preserving ancient soft tissue. The answer is fiction. Amber can preserve astonishing details, including feathers, insects, and tiny sections of skin, but it is only one pathway. Soft tissues can also be preserved when an organism is buried rapidly in fine sediment, protected from scavengers and exposed to conditions that slow decomposition. Minerals may cover or replace tissues before they disappear. Carbon can leave behind a dark outline. Phosphate can preserve microscopic details. Dense bone may sometimes act like a sealed chemical vault around material trapped inside it. Scientists have even discovered approximately 99-million-year-old bird wings in amber with feathers, skin, and tiny claws still visible. But some of the most controversial dinosaur tissue discoveries came from ordinary fossilized bones, not amber. How is your score so far? Because the questions are about to get harder. Question 4. Pay attention to this one. Scientists have detected evidence consistent with collagen in dinosaur fossils. The answer is fact. Collagen is a strong structural protein found throughout bones, skin, tendons, and blood vessels. Researchers have reported microscopic fibers, chemical signatures, and protein fragments consistent with type 1 collagen in several dinosaur specimens. These discoveries have included fossils attributed to T-Rex and the duck-billed dinosaur Brachylophosaurus. But scientists are extremely careful with these claims. Collagen found in an ancient fossil would not be a complete, pristine protein. It would be fragmented, chemically altered, or cross-linked with other compounds. Researchers must also eliminate modern contamination from microbes, laboratory materials, and people handling the fossil. That is why the strongest studies combine several different techniques instead of relying on a single test. Ancient proteins could potentially reveal details about evolutionary relationships that bones alone cannot provide. Question 5. We're halfway there. Dinosaur soft tissue has provided complete DNA that could be used to clone a dinosaur. The answer is fiction. This is where Hollywood races far ahead of science. Proteins such as collagen are generally more chemically durable than DNA. DNA begins breaking apart after death as water, heat, radiation, oxygen, and microorganisms damage its molecular structure. One major study of ancient bones estimated an average DNA half-life of approximately 521 years under the conditions examined. That figure is not a universal expiration date because preservation conditions vary, but it demonstrates how rapidly readable DNA fragments disappear compared with the age of the dinosaurs. Non-avian dinosaurs vanished approximately 66 million years ago. No complete dinosaur DNA sequence and no scientifically verified dinosaur genome suitable for cloning has ever been recovered. Soft tissue fossils may reveal chemistry, structure, and fragments of ancient proteins. They do not provide a Jurassic Park instruction manual. Question 6. Let's raise the difficulty. Iron released from blood may have helped ancient tissues survive fossilization. The answer is fact. This is one of the most fascinating hypotheses in fossil preservation. Blood contains hemoglobin, and hemoglobin contains iron. After death, iron can be released and participate in powerful chemical reactions. Those reactions may create bonds between proteins, locking them together in a process known as cross-linking. Think of it as a natural chemical fixative. Laboratory experiments using modern bird tissues have shown that iron-rich conditions can slow decomposition and help tissue structures remain recognizable for much longer. Researchers have also detected iron compounds associated with some dinosaur vessel-like structures. But iron is not a magical preservative, and it is probably not the only mechanism involved. Rapid burial, surrounding minerals, oxygen levels, temperature, and the chemistry inside the bone may all contribute. Fossilization is rarely one event. It is a chain of chemical battles, and preservation happens when decay loses. Question 7. This one catches people off guard. Microscopic pigment structures in fossilized feathers can help reveal the colors of some dinosaurs. The answer is fact. Some exceptionally preserved feathers contain microscopic structures called melanosomes. In living animals, melanosomes hold melanin, the pigment responsible for many black, brown, reddish, and gray colors. By studying their shape, arrangement, distribution, and chemical remains, scientists can compare fossil melanosomes with those found in modern birds. This has helped researchers propose color patterns for dinosaurs such as Anchiornis and Sinosauropteryx. Evidence has suggested features including dark feathers, reddish-brown tones, striped tails, counter-shading, and even possible iridescence. But these reconstructions are scientific interpretations, not perfect prehistoric photographs. Some colors are produced by pigments that do not preserve easily. Fossilization may distort microscopic structures, and not every part of an animal leaves usable evidence. Even so, soft tissue fossils have transformed certain dinosaurs from gray skeletons into patterned, visually complex animals. Question 8. Stay focused. A structure that looks like a blood vessel or blood cell automatically proves that original dinosaur tissue survived. The answer is fiction. Appearance alone is not enough. Minerals can grow into shapes that resemble cells. Microorganisms can enter fossil bones and create biofilms that imitate blood vessels or other biological structures. Modern organic material can also contaminate a fossil while it is buried, excavated, prepared, or studied. This is known as the look-alike problem. To build a convincing case, researchers examine where the structure was found, how it branches, what it is chemically made from, and whether its molecules react like known biological tissues. They may use electron microscopy, spectroscopy, mass spectrometry, chemical mapping, and carefully controlled comparisons with modern animals. Even then, interpretations can be debated and retested. That is not a weakness of science. That is science working correctly. Extraordinary discoveries become stronger when other researchers challenge them and the evidence survives. Question 9. Almost there. Microscopic soft tissue remains are found only in spectacular, perfectly preserved fossils. The answer is fiction. Researchers once assumed that possible biological remains would exist only in extraordinarily preserved fossils. But a 2015 study examined 8 ordinary-looking Cretaceous dinosaur bones that showed no obvious soft tissue on the outside. Inside, researchers reported structures consistent with collagen fiber remains, along with cell-like structures resembling possible red blood cell remnants. That does not mean every dinosaur bone contains original biological material. It does suggest that microscopic preservation may be more common and much harder to detect than scientists once believed. Museum collections around the world contain millions of fossils collected long before these techniques existed. Some of those fossils may hold microscopic information that nobody has searched for yet. A bone that looks completely ordinary could be a hidden biological archive. Question 10. This is the final question, and it's the one that surprises most people. Scientists may deliberately dissolve away part of a dinosaur bone to reveal soft tissue hidden inside it. The answer is fact. It sounds like the opposite of protecting a priceless fossil, but carefully controlled destructive testing can reveal information that no surface examination can detect. Bone contains a hard mineral component and an organic structural framework. Researchers can place a small, approved fossil sample in a chemical solution that gradually removes the mineral phase. If altered organic structures remain, demineralization can expose flexible matrices, microscopic fibers, and branching vessel-like networks that were previously locked inside the bone. Scientists do not casually dissolve complete dinosaur skeletons. Sampling is normally limited to a carefully selected fragment, and museum approval is required. Researchers must balance the permanent loss of material against the scientific value of the potential discovery. New scanning and chemical imaging technologies can sometimes reduce the need for destructive testing. But in certain cases, sacrificing a tiny piece of bone can expose evidence hidden for more than 60 million years. And that may be the greatest lesson of soft tissue fossils. A fossil is not always just a stone copy of an animal. Sometimes, deep inside, fragments of its original biology may still be waiting to be discovered. All right, how did you do? Drop your score in the comments and tell us which discovery surprised you the most. Be honest. 8 or more, you're ahead of most people. Less than that? Now you know what others don't. Hit like, subscribe, and come back for the next MTD challenge. Because the age of dinosaurs is filled with mysteries, and knowing more is how you stay ahead.